the meeting is live, and it is live. We're just hanging on a little bit for some more people to join, and then we will make a start. So hope yeah, we get started very shortly. The participant list keeps filling up, so good start. Quick thank you to everyone who's joining already, which is fantastic. And actually, I may as well say now that before we get started, if anybody wants to ask questions, then please fire them across as they come into your minds into the Q&A section, and we'll tackle them through the talks. Yeah, so you say don't hold the questions back to the end, because it might feel a little bit less relevant right at the end. And we'll give it one more minute, and then we'll kick off. Okay, so let's maybe make a start. Yeah, we've got quite a few people in sort of the audience, which is fabulous. So good afternoon, and thank you very much for joining us for the third Meaning of Light series the third in the Meaning of Light series of talks. And of course, a huge welcome to Bob and Glen. Thank you very much for joining us today. Just a little bit about the series. The Meaning of Light series is a series of talks exploring the topic of light and our connection to it. We started in September with Anne Levin, the author of Incandescent, and then last month we talked to Professor Russell Foster about circadian and biological rhythms. Next month, we'll be talking to Scott Zimmerman, who'll be joining us from New Jersey on the subject of melatonin, light, and the optics of the human body. But let's get back to today. We have the pleasure to talk with Bob Fosbury, who is an emeritus astronomer at the European Southern Observatory and an honorary professor at the Institute of Ophthalmology at UCL, and Glen Jeffery, professor of neuroscience at the Institute of Ophthalmology at UCL as well. Today, we'll be talking about the infrared revolution as we explore the subject of long wavelengths of light and the impact that they have on our physiology. Light is already an intangible element, and we are going to consider the even more intangible aspect of long wavelengths of light that sit outside the visible spectrum. I'm pretty sure we're going to touch upon photosynthesis, and we'll also explore the new term of photometabolism, and we might even touch on the subject of space travel. And before we start, we'd like to make this as interactive as possible. So please, if you have any questions, don't hesitate to pop them into the Q&A section, and we'll aim to answer the questions, as many questions as we can as they come up, rather than leaving them to the end. So let's jump straight in. Bob and Glen, would you mind telling us a little bit about yourselves? And then maybe we can kick off with you, Bob, if that's okay.
Okay, well, you've said it. I'm an astrophysicist, and by training, I retired about 13 years ago, and I've spent about 10 of those looking at Glen across a Zoom screen and or a Skype screen, which is slowing me down a bit. But I became absolutely fascinated by the interaction of light with biology. I mean, I've been working for 50 years on the interaction of light with interstellar nebulae and planets and things like that. But to begin to study the interaction of light with life was a whole new world. And I'm not a biologist. I'm not a chemist. I'm learning a lot, but hopefully not too much. I think the advantage that we have by coming in from outside the field is that we have a big picture, broader view of the subject, and a certain amount of ignorance works well under those circumstances. So we've stumbled along for a number of years looking at the already realized positive effects of light on life, and all Glen's work on the stimulation of mitochondrial function using long wavelengths of light. We've been trying to figure out how it all works, and that's what I and Scott have been doing over recent years, is to try and understand what the physics is and what's actually going on. And that has been fascinating for me, and it's been a real journey, and I must say I've enjoyed it very much, but it's at the moment we're having to talk about it quite a lot. Okay, enough of me. Thank you.
It's an amazing subject for sure. So, Glenn, tell us a little bit about yourself.
I'm a visual neuroscientist. I'm a neuroscientist that really got into vision, into visual perception. I'm not a biologist at all by background, really. My PhD was in visual bits of the brain. And I have got strong links with Moorfields Eye Hospital. I'm right next to Moorfields Eye Hospital at the Institute of Ophthalmology. And as I got older, I got interested in aging. And one of the things that regulates aging is light. So the issue of light and what light does to the body in terms of aging, in terms of metabolism, I think we started doing that about 10 years ago, and it's just exploding in front of us. I can't keep up with it. I think we're in a position now to make major advances in terms of health and well-being as we age. And the field has changed a lot as well, because instead of really talking a lot to members of the medical profession, we spend a lot of time now talking to people in things like architecture, which has been it's great for me. It's a change of the context. So work a lot with people in Bartlett School of Architecture at UCL and their environmental engineering group. And, you know, as with Bob, you know, it's taken off, it's taken over. And it's nice to know that as my career dwindles towards an end, we might be doing some good as well.
I think you'll be doing some great work, and you have been doing some great work. So I'm convinced by that. Bob, shall I share my screen for your slides?
Yes, please do.
As a starting point.
I'll give you detailed and fumbling instructions as you go
on. So here we go, and hopefully, if you can tell me if you can see.
I can see that. I can see that. Okay. Yes.
Okay. Is that good?
Yeah, that's fine.
Okay.
Before we start, I like to say I've added in the presenter notes in the PDF version of this talk. If anyone gets that afterwards, I've put some explanatory notes and a little bit of context within the presenter notes. I mean, most of those I'll actually talk about as we go through the talk. But I just want to mention that, you know, if you pick up the slides and you can't understand what the hell I'm talking about, look at the presenter notes on the PDF version, and you might get help. Okay, so I've titled this "Why Are Sunlight and Green Spaces So Healthy and Whilst Buildings Are Not?" That was the talk I used in. That was the title I used in a talk quite recently with Ulis. I think it's quite appropriate, but really, we're talking about life on planet Earth. Now, we don't know how special life on planet Earth is, but we do know quite a lot about how life operates on this particular planet. And understanding that operation is really essential for our ability to understand what's going on with light and biology, and the advantages it's giving us. So if you move to the first slide, the first real slide, Ulis.
So Bob, we've got an early starter. Phil has asked a question, if you don't mind.
Okay,
very early starter, straight in there. In what ways does modern architecture impact our exposure to natural light, and what are the potential health consequences of the reduced exposure to the red end of the light spectrum?
Well, if you wait a bit, I'll tell you.
Very early starter there, which is fantastic.
Okay. Well,
very
that's
keen.
Good.
Very
we're
keen.
We're going in the right direction. It's exactly what I will tell you. Okay. Amongst other things. Okay, so I usually start with the bottom lines. The bottom lines: sunlight powers life on Earth. I mean, that's not a trivial answer. It really does power life on Earth, and in fact, it's the ultimate super nutrient. And I'll explain. You know, in case that doesn't make any sense to you, I'll explain why that's true in a moment. But the point is that life has evolved over billions of years. I mean, the latest estimate is starting something like 4.2 billion years ago. But life has evolved over these billions of years to use light in many and subtle ways, using, and this is important, colors extending well beyond just those that are visible. We happen to see part of sunlight with our eyes. It's a rather small part. Qualitatively, it's a rather small part. In fact, we can see with our eyes the region of sunlight that actually contains most of the energy. And that makes it easier for us to see, and that makes evolutionary sense. But once we go outside that region of maximum energy, we still use the light. It's just we don't see it. And that's terribly important for this story. The next bullet here is in slightly enlarged font size, because that's the little bit that I'll be talking about this afternoon. Glen will be talking about a different aspect, the medical consequences of the understanding that we're generating and the experimental evidence which triggered us to attempt to understand what's going on. So the biosphere, and I mean the biosphere, I don't mean just humans, I don't mean just plants, I mean plants, animals and fungi. The whole biosphere is highly adapted to harvest and exploit photons from the sun. We don't see this, we can't see it because our eyes don't work in those regions where it's, in many of those regions where it's harvesting. But we realize now that that is true. And it's, you know, for evolutionary reasons, those other non-visible regions of the spectrum will be terribly important. They will be used as functional aspects of the way both plants, animals, and fungi work. Why are we studying this now? Why didn't we study this earlier? That's a very good question. And I think it's really it's a technological accident that radical changes have been made to artificial lighting over the last decades, driven largely by the quest for energy efficiency. Very reasonable desire to reduce the amount of energy we use. But in doing that, we made a very big mistake without realizing it. And we're only, you know, decade or so after that started, after that change happened, are we beginning to realize the effects on public health. I think everyone realizes that public health at the moment is not in good shape. I mean, there's an epidemic of type 2 diabetes, there's an epidemic of obesity, increasing cancer rates, you go on and on. And all the diseases of aging seem to have been affected. And what the hell is causing all this? You know, that's the big question. If we could reverse this trend, the savings in costs would be huge. I mean, the savings in costs to the NHS, for instance, could be absolutely huge. So we think it's really important. We don't know how important it is. We don't know how quickly we could change it, but we think it's really important. But it was those radical changes that have really triggered the symptoms that we've seen grow over the last decades. So it's been realized that these changes are resulting in a profoundly negative consequences for health by generating a state of infrared starvation within the built environment. Now, infrared starvation is an interesting term because we might think back to the days of sail when we sent sailors off in wooden ships to sail around the world. And by the time they got to the Cape Horn, many of the crew were dead from scurvy because they were starved of vitamin C. Now, you know, that was the reason for that was discovered relatively early, but in fact took the British Navy about 100 years to make a change and give them lemons. But it turns out that this 21st century scurvy is not the same cause as vitamin C scurvy, but the symptoms are not dissimilar. And I think fundamentally the biochemical reasons are not dissimilar either. And the interesting thing about it is the timescale of development of these symptoms seems to be rather similar. That is, you know, if you sit in the dark all day, you don't suffer immediate symptoms. But if you sat in the dark for weeks and months, you would. And one of the triggers that helped us understand what was going on was the recent discovery that all the astronauts in the space station who go up for 6 months or so on the space station, both the astronauts and the experimental animals that have been sent up to do experiments with, they all suffered a decay in health, which was the common element of which was mitochondrial dysfunction. That means the energy generating organelles in the cells were not operating properly, and they were causing a range of symptoms, which include pre-diabetic symptoms, and so on. And we realized that, if you're in the space station, ironically, you don't see the sun. I mean, they don't let the sun shine through the windows of the space station. So you're basically in an all white LED light environment. The lighting was changed in the space station about, I think, 12 or so years ago, from a mix of incandescent and fluorescent to pure white LEDs. They're circadian adapted to give some circadian cycle to the astronauts, but they're all white LEDs. And as we all know, white LEDs don't emit any near-infrared radiation at all. And so for us, it was, fairly obvious that a major cause of this health problem on the space station was the near-infrared starvation. And actually, that opinion appears in a recent report, which has been given to all the space agencies around the world from the Gaia Foundation, which is a quantum biology foundation in Britain, which is gathering together quantum biologists from around the world. And they wrote a big report on space health, which came out just at the end of October. And that's having an impact on the thinking of the space agencies in terms of space flight. So those are the bottom lines. And now I'm going to go through and I'm going to explain some of the things that we've done and what they might mean. So if you could move on to the next slide.
So before we move on to the next slide, Bob, we have another question, but I'd like to maybe just come back to Phil Clark's question about modern architecture and the impact. And when we talk about the space station, our, let's say, modern indoor environments, when we merge that into the sort of knowledge that we have about the amount of time we spend outdoors, which is roughly 90% of our time is spent indoors, and if we're spending a time, if we're on the American side of the Atlantic, then that's 93% of our time indoors. Actually, there's not that much difference between the space station and our modern indoor environments. The only difference really is that we have a choice, and the choice is to step outside or not, as the case may be. That's how it feels to me. Just suggesting that it's not that different.
No, it's not that different, but actually, I mean, the space station is different in that they have zero gravity, and that has effects on metabolism and health as well. So the point is that in the space station, it's not so obvious what the major cause is. I'm sure that the infrared starvation is one of the causes, but we're not sure that it's the most important one. So the obvious thing we could do then is to look at ground-based environments that are similar to the space station in terms of lighting. And you immediately think of ships, submarines, and so on, where people are in artificial light sources for weeks or months at a time. And if we could get hold of data for those situations, hospitals and intensive care units as well, obviously, if we can get data from those environments and compare those data with the space station, we could begin to disentangle what the various causes were. So that would be a very useful thing to do.
Thank you. And then Radim has asked the question as well. Yeah, it would be nice if we could comment on the differences between our demands on NAR in daytime and nighttime.
Well, I think that the demands are naturally very different, because at nighttime you wouldn't have any lighting, or you wouldn't have any NIR, unless you were sitting around a campfire, and that's another story. But so the demands on near-infrared radiation seem to be linked into the circadian cycle. And as Glen will explain later, the most beneficial effects of adding near-infrared radiation are in the morning after you wake up, when your mitochondria need to stop all their housekeeping that they were doing overnight and start generating energy for real. So yes, it's a link to the circadian cycle there.
Brilliant, thank you. So I'll just split the slide for you.
Yes, this is a phrase which is relatively easy to remember and really encapsulates a lot of what we learn. Life on planet Earth is an antenna. Go outside and look at a tree in the summer. It's an engineering structure to hold leaves and point them towards the sun, as are many plants. So life on Earth is an antenna, basically, to collect sunlight. And as I said before, it collects visible sunlight to do photosynthesis in plants, but in other forms of life on Earth, it will collect and harvest other wavelengths to do other things. So it's an antenna with receivers tuned to sunlight, and importantly, and not so obvious, and radiators tuned to the cold of outer space. So what happens when you absorb sunlight into either a plant or a body? That sunlight will get processed by wonderful mechanisms that have evolved in plants and animals and fungi to utilize life to do stuff. And in plants, you utilize life to do photosynthesis, which is actually to generate carbohydrates, which actually are the source of all our food and most of the energy that we use on the planet. So clearly, photosynthesis is an important process. It's been known about for 250 years, but the details have only been sorted out in the last few decades by physics. Physics have understood the details of photosynthesis because it's highly quantum mechanical. It is quantum biology. It's pure quantum biology. But once the energy of the visible photons in photosynthesis are converted into carbohydrates at some efficiency. It's not terribly high, but it's high enough to work. And so it's the solar photon energy that's used to make the food. And that's an important point to make. It's a direct transition between solar photon energy and the energy contained in sugars. Now, when I come to talk about the infrared in bodies, it's a different process, and it's different in an important way, but I'll explain that a bit later. But the point of this slide is to say that when the energy, the photon energy, sort of flows down through your body and performs all these functions, you'll eventually have used up all the useful parts of that energy, and the waste product will come out as heat. So it's your body is a bit like a gut. The mouth of the gut is your skin, your integument, and that soaks up the sunlight, and you excrete the used solar photon energy by radiating that energy out into the room or into the outside world. The outside world is much better for reasons that I'll explain. But it's a process of metabolism. I mean, you eat food, you metabolize it, and you excrete the contents of some of the contents of the gut. Here, you're eating sunlight, you're digesting it in the body, and you're excreting the waste product, heat and disorder, out into outer space. So very close analogies in these two processes. And that's why we've invented this term, or you utilize the term photometabolism to represent what we're talking about. It's the mirror of photosynthesis. It's photosynthesis takes sunlight and makes food. Photometabolism takes the food, metabolizes it, and makes energy which you can use to run around and think and do all the things you do. So this phrase represents the whole process from beginning to end. It's an antenna, you collect the sunlight, and you do what you have to do with it in your body, and then you radiate it out to outer space. And I'd like to make a point here that, I think if you ask anyone in the street, why do you eat? They may say because they enjoy the restaurant down the road, but actually they probably say you eat to make energy. Well, in some sense, that's true, but really you eat to keep your body in a high state of order. The functions in your body are highly ordered to maintain your temperature at a constant temperature of 37 degrees centigrade and keep you well removed from a state of thermodynamic equilibrium. If your body were in thermodynamic equilibrium with your surroundings, you'd be dead. In fact, that's the definition of death in some sense. So the food the nutrients that you absorb are to keep your body operating properly and enabling you to do things. So photons are a metabolite exactly like the food you eat. There's very little difference in function. They're combined together to give you the energy, but both light and food are nutrients. And we've heard a lot about high quality nutrients keeping you healthier. All these nutritional podcasts that you watch on the TV telling you to eat proper food and not ultra processed foods. That's because high quality foods are highly ordered foods. They come in, when you eat an apple, it's a highly ordered structure. And that structure enables you to digest it in the right place in the gut. And so while it contains a lot of sugar, that sugar is much better for you in the form of an apple than it is in the form of a sugary drink, for instance, which you would metabolize immediately. So, the good foods are the highly ordered food. They're structured and highly ordered. Photons are a fantastic form of energy because they're almost zero entropy. Entropy means disorder. They're highly ordered forms of energy. And so they're a very, very, very high quality nutrient to go with the high quality food you're eating to keep you healthy. I can't emphasize that too much, really. Photons are a nutrient and they're very high quality.
Brilliant.
Okay.
We have a couple more questions, actually. One from Martin Moore-Reed. And Martin, I've just finished your book and I've thoroughly enjoyed it, so thank you very much for that. So Martin's he's put also we have ultraviolet and violet light starvation as well as infrared starvation in modern buildings with UV and IR filtering window glass, which I think we've discussed before on many occasions.
That's absolutely right, and I think we're certainly not arguing only for the re-imposition of the infrared. We're arguing for the whole solar spectrum, and I think we talk about infrared for reasons I'll explain, which is because it gets deep into the body, which is very special. But the longer wavelength infrared, going right out beyond 2, perhaps up to 3 microns, where the sunlight becomes very weak and becomes overtaken by the energy radiated from the Earth. The whole of the solar spectrum that reaches the ground, we think is important for functions within the body, not necessarily only metabolism. I mean, as we know, the ultraviolet is very important for vitamin D synthesis and other things. But so really, it's the whole solar spectrum that we need to reinstate in some sense. And that's why we recommend people go outside. It's harder to put UVB radiation inside a building. You can, but all the pictures would fade and the carpets would fade, and people would get very upset. So yes, you can do it inside buildings. And in fact, in the old days, they used to go around care homes, didn't they, and put up UV lights and put goggles on the old age people in the care homes. But they don't do that anymore. But they were doing that. So I agree. Not only infrared, but certainly UV. All the wavelengths are important. It's just the ones that we don't know very much about are the infrared at the moment, and we're trying to figure it out.
And then one more from Morali, who says, please forgive me if this question has already been asked before. Just learning about all of this as of yesterday. The question he has is, how does the role of infrared light compare to other parts of the light spectrum in supporting human health? And could there be a trade-off in focusing on infrared exposure over full spectrum sunlight?
I will talk about that. won't talk about it now. I'll come to it. I mean, it's a very good question, but it's we know more about some regions of the spectrum and their biological implications than others. So we have to be very careful in answering that question, I think. That's all I'll say at the moment.
Okay, thank you. Shall we move on to the next slide?
Yep. So, yeah, you've gone too far. Can you go back a couple
sorry. There we go.
Yeah,
there we go.
This got, yeah, first build. That's it. Okay, this has got several builds in it. So this is a spectrum of sunlight. I hope a lot of people are familiar with looking at a spectrum, but basically the y-axis, the vertical axis, is the brightness. And here the brightness is expressed in something called solar irradiance in rates of photons, photons per second, per square meter, per nanometer. Photons are the important things for biology. It's not so much necessarily the energy you're getting, it's the number of photons, because it's the photons that interact with molecules. So we tend to use spectra expressed as photons rather than watts per square meter. I mean, they look very similar, but actually, because you multiply by wavelength to get from energy to photons, everything's sort of biased slightly towards redder wavelengths. So if you compare this to a picture you see in the book, it'll seem sort of slightly moved over to the red end. The blue line there is the light that comes from the sun outside the atmosphere before it comes through the atmosphere. And the orange line there is a typical spectrum of daylight with a solar altitude of, I think it's about 30 degrees. Oh no, I beg your pardon. It's around 40 degrees. It's 1.5 air masses. That it's you're coming through twice the amount of you're coming through 1.5 times the amount of air you'd come through if the sun was in the zenith. And you can see the difference between the blue line and the orange line as well. The orange line is lower because some of that light has been used to make the blue sky. But as you go to redder wavelengths, there are strong absorptions. The downward sloping lines here. You can see my cursor here. You can see my cursor? Yeah.
No,
Anyway.
I'm showing it using my cursor, so hopefully people can see it there.
The strong lines here are due to water vapor in the atmosphere. Okay, you have water in your body as well, and there are similar absorption lines in your body due to water, but they look rather different because the water is liquid and the water vapor in the air is a gas. But there are a few other lines due to the oxygen molecule and so on over here. But basically, most of the absorption bands are in the infrared, near infrared and infrared parts of the spectrum. Now, down here in the ultraviolet end of the spectrum, below 300 nanometers, you don't get any light through the atmosphere at all. It's completely absorbed by ozone. But the little bit just beyond 300 nanometers, you get a little bit of the light through the atmosphere when the sun is relatively high in the sky. And that's the light that's used to convert cholesterol into the precursor of vitamin D. And make this essential hormone in the skin. I'll say quickly that there is an interesting thing here is that we generally think when the sun's low in the sky you don't get any ultraviolet UVB radiation and you can't generate vitamin C. In fact, you can get quite a lot of ultraviolet radiation from the sky because the sky is coming from light that's traveled high through the atmosphere and then bounced down towards you from above your head. And that travels through a much shorter distance through the ozone layer than the light that's coming directly from a low sun. So in the winter, you can actually get a lot of ultraviolet radiation from the sky. But the trouble is you're wearing clothes which absorb it. So, you've got to be naked in the winter to get benefit, which is not it's not very easy unless you happen to be a Swedish sea ice skater. They get lots of vitamin D in the winter. Now, the vitamin D, is in the ultraviolet. Most of the photosynthesis is driven by blue light and red light together. Green light is not very effective for photosynthesis. That's why leaves are green. It's the waste light that it's not easy for photosynthesis to use. It can do it using other pigments, but most of the photosynthesis happens in the blue and the red. But it doesn't happen in the infrared. There are no, except for some bacteria, there are no absorbers of light in the infrared for doing photosynthesis. And so I've also marked on this baseline plot the emission from a white LED, the green dashed line down the bottom on the left, which you can see is entirely restricted to the visible part of the spectrum. Now here's a tungsten filament lamp here, which goes way up in the infrared. And this is why we've got rid of them, because they emit so little light and they emit so much infrared light that you were spending all that electrical energy in generating lots of infrared and very little for visible light. But, and can you move to the next build, please?
Yep.
I've changed the shading on the plot. It's the same plot. I've changed the shading. The red over here represents the absorption of light, visible light in the body, principally by hemoglobin. Your blood is red, and it absorbs blue and green light. So any light entering your body in the visible part of the spectrum will get absorbed relatively quickly by blood and other pigments. But it won't penetrate deeply. When you move to the other end of the spectrum here in the infrared, you've seen already these water absorptions I've shown you in the spectrum, down here. Light is increasingly absorbed by water, not only in the atmosphere, but also in your body. And there's a region in the middle here, which is labeled tissue transparency window, which is a well-known biological phenomenon. You find it on Wikipedia, and you can look it up. It's the region where the body is relatively, I won't say transparent, it's actually translucent, because the body in those wavelengths, not only your body, but also fungi and plants, they're all highly efficient scatterers in this region. They don't absorb light very readily, but light can come into your hand, for instance, instead of being absorbed a millimeter into your flesh, it'll be bounced around by a cell wall or something. It'll be scattered, and it can scatter many times in the body before it hits an absorber. And this makes this infrared window unique. It's the only spectral region where you're almost completely dominated by scattering in the body, and it allows the photons to penetrate deeply, and as Glen will show, right through your body. You can shine light right through your chest, in fact. And that's incredibly important, because it means essentially all of the mitochondria in your body, which we show are sensitive to infrared light, can access this through your skin. You don't have to plug in fiber optics and shine infrared light into your body. It'll come through the skin. So this transparency window is incredibly important for the functions that the mitochondria give you when they're provided with infrared light. So photometabolism. And if I give me the next build, you'll see a grey line here, which is it's not an intensity, it's a transparency window. So the transmission at the top is good, and the transmission at the bottom is poor. So this is a window of transparency, the grey line there, which is actually a measurement of the transmission through my hand. Actually, it's through my, I think it's through my right hand, actually. Yes, it's through my right hand. So it's about 3 or 3.5 centimeters of flesh the light is getting through, and I'm measuring the spectrum. And you see this peaks at around about 800 nanometers. And that means that, the light is scattered many times before reaching that point in the middle of my hand. And the consequence of that is that the light has actually spent more time in my body than it would if it went straight through, because it's traveling a longer distance. It's doing a random walk. It's like a drunken person in a car park stumbling from light fitting to light fitting, from lamppost to lamppost. They do a random walk. And the mathematical properties of a random walk is if you leave this random walk going, the distance you travel is dependent on the square root of the number of scatterings. So you'll do a random walk, but you will always travel some distance, and on average, you'll travel a distance which is the square root of the time of the routes you've taken between scatterings, and so that means the photons spend a long time in your body, and that means that the photons coming into your body from outside actually build up inside your body because they're coming in at the speed of light. And then they're traveling through your body at effectively much slower than the speed of light. And so the density of photons builds up. Although not all the photons, only about half of the photons will enter your body, but the ones that do will build up in intensity. That means your whole body glows in the infrared. We can take photographs of this. All of life, if you shine infrared light on it or white light on it, it will glow in the infrared. And so these photons very efficiently get into the body. And there are many subtleties in the way that it does this. And Scott Zimmerman, who will be giving the next talk here, has pioneered this work on penetration into the body. And it turns out that all life is highly adapted, as I said in my bottom lines, it's highly adapted to getting light deep into the body. So this region, this transparency window here, is the region where photometabolism happens, and that makes it special. The light in other places will do different things, probably. It may contribute to this, but it may do different things. But I think the photometabolism happens mainly during in this window of tissue transparency. Now, the next build is a shocker because it makes it graphically very clear what we've done over the last couple of decades. And what we've done is removed everything except this white region in the middle. The LEDs you can see, and this is the kind of transmission function you get with a modern glass, which is specially made to exclude everything but the visible. So it's a mirror of it follows the spectrum of an LED very closely. And you see we've basically cut everything out of this transparency window here. And, frankly, the health effects of this are devastating, we think.
So that's sort of... Sorry, I was going to say that sort of picks up on what Martin Moreed was pointing at, in terms of, even with glazing, we've narrowed our access to some of the spectrum.
Yeah, we were talking to one of the major glass manufacturers a few months ago, and I asked him to put up the transmission curve of their best-selling glass, and he proudly put up this one. And I couldn't stop myself. I said, but that's a disaster. He wasn't listening before I said that. He listened to the rest of the talk, I think. Yes. Do you have some other questions, don't you? Because flashing on my screen.
So there's a question from Joe Vose. Hi, Joe. Do you think the concern around skin cancer has played a role in the minimization of non-visible light in standard light sources? So can I just jump in there before you pick this up, Bob? We've been talking to Professor Richard Weller, and I believe, Glenn, you've met Richard now, and he is a leading dermatologist, and hopefully we'll be talking to him early next year, because talking about skin cancer and exposure to full spectrum light, as in daylight, and the fact that the narrative seems to be changing within the field of dermatology, and related to all-cause mortality. But that's something slightly different. But back to the question that Joe had, do you think the concern around skin cancer has played a role in the minimization of non-visible light in standard light sources? Is that a thing, do you think?
Well, I'm not sure it is for light sources so much, but it is for sunlight, I think. And I went to a dermatologist recently, and he is also a dermatologist who very much favors getting people out into sunlight, basically because he's read Richard Weller's paper. And but he's an absolute convert, and we actually, he's coming to visit me quite soon, and we're going to discuss all of this. But yes, I think the thing we're saying about getting out into sunlight, and Scott says this relentlessly, we're not saying to get out into sunlight you should go out and spend all day on a sunbed slathered in sunblock and try and get a suntan. You know, there's responsible getting out in sunlight. You know, if you wear clothes over most of your body and you wear a hat and you're out in direct sunlight, you can protect yourself from the UV very effectively, but you'll still let the infrared through. So you'll get the advantages of the infrared without the dangers of the UV. But yes, I mean, my answer would be based on what Richard Weller says. Basically, yes, you need all these parts of the spectrum. I'm not sure if I've answered that question precisely, but.
Well, yeah, I think it's probably quite a tough question to ask in terms of interior spaces, but certainly an easier question to answer in respect of, you know, the full spectrum of light.
Well, I'll tell you what I do, because I mean, I use the infrared light in the morning in our bedroom. I switch it on when we wake up, and I leave it on for 15 or 20 minutes until we get up. Also, I've started using a fluorescent UVB source, which is used for reptiles. If you're keeping reptiles in a tank, they need UVB, and they make these special UV phosphors to give them UVB. And I've started putting that on for 10 minutes in the day as well, as an experiment. So there are ways of bringing UV inside. You'd probably do it with LEDs, but I think it's a complex question to answer. How could you do that in public spaces healthily and safely? But yes, the ideal lighting indoors would include the UV for some reasonable period of time, short period of time, presumably.
Fantastic.
Okay. Next one, please.
Here we go.
Here we go. My the most viewed picture of a hand in history, perhaps, except for Leonardo da Vinci's. Yeah, it's this was a surprise to me. Apparently, the medics say they knew this. They're shown this at medical school, but I don't think they interpreted it as being anything important, perhaps. But your 850 nanometers, your flesh is translucent, and there are very few absorbers there. For instance, you don't see the bones there at all. The bones don't absorb; they scatter perfectly well. So they're as efficient at scattering as the tissue is. But if you go to the next build of this, you'll see the other side of my hand, or you see my left hand. You see the veins. You know the veins are on your hand or on the back side, very close to the skin. And they're the major absorbers of light in human tissue, and it's the deoxygenated blood there that has a significant absorber at about 850 nanometers. I can't remember exactly what it is, but it's on the plot somewhere. And I think there are other absorbers which are probably much more important for mitochondrial function, but they're all weaker than that, and we don't see them very easily. But they're there, and the photons that get into your body will find them and get themselves absorbed. And we think that's what's that's the key interaction between the photons and the biological tissue. And the phenomenon you're seeing with my hand, it's interesting that if you put your finger over a white flashlight, I'm sure you've all done this, it looks red, doesn't it? And you think immediately, oh, that's because of the blood. But actually, it's only a little bit due to the blood. The red that you're seeing by shining light through your finger is analogous to the red of the setting sun. It's all to do with scattering. And I can put a perfectly white object on a source of light, and it will glow red if it's a living object. And I think I've got one in a moment
Yes,
to show you. So why don't you put it on?
So I'm going. So Simon Carter has just posted something in, and Simon is the author of Rise and Shine. And if I remember rightly, it was around. I think Simon wrote the book around 2007. And I read it recently, and it's a fabulous sort of sociological study about our connection to light. And he's just posted that in the 1920s, there was a product called Vitaglass that allowed the full spectrum through glass. Unfortunately, it degraded quite quickly. So, we had products in the past, and we knew about this sort of stuff. We seem to have moved away from it. So I'll move across to the next slide. There we go. I did wonder what this object was actually. Is it a mushroom of some kind?
It's a lion's mane mushroom. They're supposed to be extremely healthy. They're for your brain and so on. And they're called lion's mane mushrooms. And we have a fungal person at the market at the weekend here in Bath, and I buy lion's mane mushrooms from him. This is a slice off the end of one of them. It's about 1.5 centimeters, 2 centimeters thick, and it's absolutely white. And I put it on my, I have a spectroscopic integrating sphere here, which is a very bright tungsten filament lamp shining through a hole. I just put this on top of the hole and it glows red. So where's that red coming from? Now, where's the red of the setting sun coming from? The red of the setting sun is because all the blue light has been used to make the blue sky when the sun's low in the sky. So you're only left with the red light. There's no pigment there at all. It's just a product of scattering. In fact, I have objects which are made out of glass, which is a perfect scatterer. And I should have put them in the view graphs here, but I didn't. You can see, I call him Rayleigh's fish. If I shine light through the eye of the fish, it sorts the photons into order for me. It scatters the blue light first, then it scatters the green light, then it scatters the yellow light, then it scatters the orange light, and it lets most of the red and the infrared light through. So if you have a perfectly scattering medium, it will sort the light for you. And what's happening here is you're scattering the blue light out first, so it doesn't come through, but you're leaving the red light at the end. And this is what happens in your body. You get rid of the blue light at the surface of your body, but it's the red light and particularly the near infrared light that penetrates deeply and gets in. And so this scattering dominated structure that you have when you have no pigments or very few pigments is so important. And this is why leaves are thin, because the photosynthesis uses very strong pigments, uses chlorophyll. And so you have to make a thin leaf in order to get the light into the chlorophyll. And because the plants don't need infrared light, in fact, it's damaging to them because they would get hotter and they would radiate water and lose water more quickly. They scatter the light very, very strongly in the infrared and get rid of it. And so if you sit under a tree, you're being illuminated by a surface which looks kind of dark green, but actually it's brilliant red. It's brilliant infrared. And a mushroom, which normally grows in the forest understory, will accept this infrared light. It will scatter it through its tissues into the spore-producing regions, which are 1 centimeter or so below the surface, and use that infrared light to stimulate the mitochondria to give them enough energy to build spores. And I think this is a very important realization. Reproduction is a very energy-intensive process, and it turns out that all of life, fungi, seeds, nuts, fruits, they're all highly adapted to be scattering dominated and getting the infrared light deep into their core to illuminate the seed that is trying to mature. And this is true for animals, humans. A woman's skin gets stretched during pregnancy, and the skin becomes highly transparent to infrared radiation, and the fetus needs infrared radiation to develop properly. It's designed that way by evolution. And so all of reproduction in the whole of the biosphere is actually driven by the presence of infrared light accelerating the metabolism, giving you effective photometabolism. It's a very, very fundamental process. But I just thought the bathroom was a rather beautiful illustration. I have many other examples. If you take a thin sliver of wood, a few millimeters thick, and put it on a bright white light source, you'll see red light coming through. If I put an olive on my on this thing, it will glow bright red. Insects, I put an insect on an infrared light source, and it just explodes in light. You know, invisible light, it's black, it just shields you from everything. I've got a picture of this on the web somewhere. If you shine 850 nanometer light on an insect, the whole thing glows. It's quite remarkable. So there's a radical change in the behavior of light in tissue between the visible and ultraviolet, where you have lots of pigments, and the near infrared, where you have very few pigments. It's a completely different it's a completely different behavior of light, multiple scattering and more subtle processes where you actually trap the light with nanostructures inside the body. You know, the nanostructures that make butterfly wings, brilliant colors and so on. Those appear all through life in plants, animals, and so on. They're inside our bodies as well, and they're trapping light to enhance metabolism. Okay, let's move on to the next one. I don't want to go on too long. Tell me when I should shut up. Anyway.
Maybe so we give Glenn a bit of time.
Yes, exactly.
Maybe
yes.
sort of another 5 minutes. Is that good? Do you think?
Yeah. Okay. Fine. I'm nearly at the end now. Well, this shows a scene in a wooded landscape in the infrared, taken at about 800 nanometers wavelength, and you can see all the leaves are incredibly bright. And in fact, the shadows under the trees are actually less dark than the shadows would appear in visible light, because they're getting the infrared light bouncing down into the shade of the tree. So, you know, when you're walking around in a forest, you see these walls of brilliant near infrared radiation. So it's incredibly healthy to be outside amongst trees and to be under trees. And if you want to shield yourself from too much ultraviolet light, go and sit under a tree, and you still get all the advantage of infrared light. Okay, that's enough for that one. And this is just to show that your clothes also are generally pretty transparent in the infrared. This is a bunch of clothes. It's a woolen jumper, a shirt, and a t-shirt. So 6 layers here. This is with 660 light behind it, 660 nanometer light behind it, taken with a visible camera. And this is with 850 light behind it, taken with an infrared camera. And you can see that the light gets through the clothes very easily. It actually gets through your body relatively easily, but not quite so easily as well. But so you don't have to take your clothes off to get the benefit of infrared when you go outside. And clothes will effectively protect you from overexposure to UV. Okay, next one. This is a science slide, and before I take you through it, I'll just tell you the basic principle here is we have a body, a human body at 37 degrees centigrade, and it's full of molecules, and those molecules are bumping around amongst one another because of the kinetic temperature of the body. 37 degrees, they'll be bumping into one another with a given speed and energy, and that means that a molecule inside your body will have an average energy associated with that temperature with that motion of the molecules, and that's characterized in physics or in chemistry as kT. It's Boltzmann's constant times the temperature, and the actual the energy contained in that molecule is 3/2 kT, and that's quite small. And in order to perform some functions in your body, it's very hard to perform those functions without climbing a hill first and getting over it before you can actually perform the function. And you have to wait a little while for that to happen. And that's what happens in the body. You have a molecule here sitting at the bottom of a hill. It needs to undergo a transition, which actually would produce energy. It goes to a lower energy level down here. But in order to do that, it has to go over the hill. And it only has 3/2 kT, which is a relatively small amount of energy, which makes it difficult to climb the hill. Now, how does it manage to climb the hill? It can do it infrequently by quantum tunneling. This is a quantum mechanical effect where you can actually push that molecule, burrow it through the hill to the other side using quantum tunneling. And that's well known. It happens in biology all the time. But if I introduce a near-infrared photon into that picture, which you do by pressing the next button, this near-infrared photon is coming from the sun. Now, we think of near-infrared photons as being relatively low energy, but actually a photon at 800 nanometers has 40 times the kinetic energy of motion of the molecule. So if you represent the molecule as worth a pound coin, the near-infrared photon is worth 40 quid, two 20-pound notes. Interestingly, it's Turner on there talking about light and color. I thought very appropriate on the 20-pound note. But there's a huge energy advantage if this molecule absorbs this photon. So if you press the next button, you've suddenly given this molecule 40 times as much energy without heating it up. You've excited it to an excited state, a quantum mechanical state. And the temperature is the same. It still has the 3/2 kT of kinetic energy, but it's got a couple of 20-pound notes to spend, and it can spend that on whatever it wants, but it may want to spend it by reacting with another molecule next door and climbing this mountain and getting over the other side. And actually, if you press the button, it'll zip over the other side with no problem. And you'll end up, if you press it again, you'll end up with the answer that you wanted. And so once we introduce the idea of having photons inside the body, and we think of the body as being in a stable state at a stable temperature, and that stable temperature is capable of performing all the biochemistry that you need in darkness. If you walk into daylight, you suddenly introduce this field of radiation inside the body, which is a relatively high energy field. And although the photons are relatively rare compared with numbers of molecules, their energy is so valuable, and they can accelerate all kinds of functions. And this is the way we think the infrared photons interact with the mechanisms inside the mitochondria that are responsible for making the energy that the cells use. So it's a huge advantage to the mitochondrion to be in infrared light, because it can perform all these functions very easily with the energy that it has. It's not using all that energy it's not like photosynthesis. It's not using the energy to make the product. It's using the energy to catalyze the product. So it's basically oiling the engine. It's oiling the mitochondrial engine and allowing it to work much more efficiently and faster. Okay, enough for that. And we're nearly at the end now. So there's a nice cartoon I put together for sort of encapsulating what we've been saying. If you look up at the top left, you see the sun. The sun's nearly 6,000 degrees of centigrade or kelvins or whatever you want to use, but it's getting on for 6,000 degrees. And that's radiating down onto the ground and the tree here. And in the leaves of the trees, it's performing photosynthesis, which will be making sugars, which in principle we're able to eat. I use a tree because it's easy to envisage, but it could be a stalk of corn or whatever you want, an apple or whatever you want. So you have the sunlight at 6,000 degrees coming down, using its photons to make the food that we eat. Meanwhile, the tree is scattering the the infrared radiation, which the photosynthesis doesn't use, down onto the ground, and all around, in fact, but down onto the ground. And this couple who are sitting under the tree in the shade, they can use this infrared radiation to help metabolize the sandwiches they're eating at the time. And they're comfortable sitting under the shade, and they're eating their sandwiches, and they're performing photometabolism. They're digesting it very easily, and they feel comfortable because they're not roasting under the sun. They're able to radiate the energy that they're excreting from their bodies. They're able to radiate the energy into the clear blue sky. Outer space is at minus 270 degrees centigrade, but the sky, because of water vapor in the upper atmosphere, is at about minus 40 or minus 50 centigrade. But that temperature difference between your body and the sky is very much bigger than the temperature difference between your body and the wall of a building. So the second part of this cycle, where you're getting rid of the excess heat from your body that's come from the metabolism, is much more effective when you're outside. And I think under some circumstances, this is important. It certainly makes you feel good when you're sitting outside in the shade of a tree.
So, it does.
Yeah, so we all know this and we all understand that. But, and the thing is here, you have a process going on in life, in photosynthesis in your body, highly complex biochemical process going on, which is using photons. Photons is the energy source which is driving those biochemical processes. And you're taking photons from a very high temperature. They're very dilute. There aren't many of them when they reach you, fortunately, otherwise you'd vaporize. They're very high energy photons coming into your body and giving you extra energy. And you're able to radiate this energy into outer space. And what does that remind you of? It reminds me of a classical heat engine, a Stirling engine. So if you go to the next picture, you'll see a Stirling engine, which I built with my grandchild out of a kit. It was great fun. So this Stirling engine is being driven by the difference in temperature between the top plate and the bottom plate. The bottom plate is sitting on an ice pack from the freezer. So there's a significant temperature difference between those two plates, and it's driving the engine. Now there's a very close analogy here between this and life. Life is instead of using kinetic energy, we're talking about temperatures here, kinetic differences in kinetic energy level. In life, we're using the difference between the temperature of the sun and the temperature of cold of outer space. So between 6,000 degrees and minus 270 degrees is being used to drive life. And that is what life does. You remember life is an antenna tuned to sunlight and radiating to the cold of outer space. It's driving a heater. You're the heat engine here. You're much more complex than this, but your life and the life of a plant, the life of a fungus, is basically a heat engine. Okay.
Next slide.
Finally, I just wanted to make one or two suggestions about what could we do about this and what might lamps look like. This is not a problem that I'm addressing. This is the problem that you're addressing. This is just the ideas that I guess both Scott and I have. What might the lamps that we evolved from this discussion, what might they look like? Well, I think the visible light will still be from white LEDs, hopefully warm ones and not cool ones. You don't want too much blue. The infrared, it's very hard to avoid having filaments at a lower temperature than Edison bulbs. This makes them very different from Edison bulbs. They run at a lower temperature. The filament doesn't get so hot. It never breaks. It never blows out like the old tungsten bulbs used to after a year or so. They run at a lower temperature. That means they use lower power. They have a lower color temperature. They have very long lifetimes, and they're essentially 100% efficient at turning electricity into heat energy. But when you do that, if you go to the next slide, how do we fill the gap around 800 nanometers? So the next slide is again the spectrum of the sun in blue in this case, and the spectrum of a white LED, a warm white LED. This is actually the one from your lamp, Ulysses.
Okay.
And the red is the combination between the white LED and a 2500 Kelvin glowing filament. This is a couple of 100 degrees cooler than, you know, a normal quartz halogen lamp or something like that. And this provides plenty of infrared energy. It's much weaker here because we're running it at a lower temperature. And you know the total brightness of a hot body goes as to the temperatures of the fourth power. So if you drop the temperature a bit, you reduce the power. And when you combine those two, you cover pretty well the solar spectrum. You don't cover the ultraviolet, of course, with this. You could think of other ways of producing the ultraviolet, but you leave this rather embarrassing gap in the region where you want to do photometabolism. So, you know, the question I would pose to you guys who are thinking about light sources is how would you do that? And if you press the next button, I just give an example of, you know, one thought about how one might do that. I just took a couple of 800 nanometer LEDs, one at 810 and one at 850, and combined them together, and they fill the gap. It's not a very tidy solution, but they will fill the gap. So here we get a relatively simple lamp, which produces a pretty good match to the solar spectrum. Okay, and my very last slide, which looks really scary, I apologize for that, but I'll explain why. Well, that's just what I've said in words. You can read that afterwards. I won't bother to go through. I've already said that. So the next one, please. I put this up because it really contains the essence of what we're talking about. I put two mathematical equations up here. Some of you will recognize them, others will not. One to do with light, and this is what's called the Planck function. This is how Max Planck, this is the equation that Max Planck derived late in the 19th century, and it was the beginning of quantum mechanics, basically. He didn't understand the physics of this at the time, but he guessed at the form of the equation, which turned out to be absolutely spot on. And the point about this equation is it contains both classical thermodynamics, the temperature, and Boltzmann's constant here appears in this equation, which is a representation of classical physics, classical thermodynamics, pre-quantum mechanics ideas, all the 19th century physics. But it also contains Planck's constant, h, and the frequency of light. H nu is the energy of a photon. So light is represented by an equation which combines classical and quantum physics to make the origin of the light that we see. Now on the bottom here, I've got an equation which similarly represents life. This is actually the solution of Schrödinger's equation for quantum tunneling. And we know that quantum tunneling occurs in mitochondria. And similarly, we have this combination of quantum mechanics, the H, and the E here is a photon, is a quantum mechanical energy. That's and the other energy here is sorry, this is the quantum mechanical energy. The E is a kinetic energy here. So both of these equations in light and life involve a mixture of quantum physics and classical physics. So anyone who suggests to you that quantum mechanics is not relevant to biology needs to go back to school.
I'm not going to lie, the equations are quite a tough one for me.
Well, you don't have to understand. You don't have to understand that one of these equations represents light, and the other equation represents the process of mitochondrial function. Doesn't matter what they are. So don't try and understand what they are. It doesn't matter. It's simply that there's a combination between the two. I've shut up now. Okay.
I'm so pleased I don't have to pay too much attention to the equations. But thank you very much, Bob. Glenn, you've been super quiet, and maybe if you want to jump in and share your screen and talk us through the next phase of the talk.
Okay. Yes. Can you see my screen?
I can see your screen. And you're just about to make it bigger, which is even better.
I'm just about to make it bigger. Okay, all right. So what I'm going to talk about is I'm going to talk about why different spectra of light that we experience, both outside and in the built environment, are important for our physiology. And I'm going to talk about ways in which we can correct some of the problems that we've got in terms of physiology due to light in the built environment. And I'm going to be talking about these guys here that are mitochondria. So I apologize to some of you who've seen these slides before. Hopefully I've chucked in a few that are slightly different. So mitochondria, they make ATP. ATP is the energy that drives all of your body, and you make your own body weight in ATP every day. The mitochondria are unbelievably important. As an example of a mitochondria, there's about 1,000 of them, we think, in each cell, but it changes by the minute. They control your aging. That's absolutely critical. If they're annoyed, you'll die relatively early and you'll be unhealthy. If you maintain your mitochondria in a relatively good state, you'll live longer and you'll have a longer health span. Mitochondria are very interested in light. They're watching what happens to light, but we're going to talk about them just as if they're a battery. Okay, so they promote health, they drive disease, and they can tell you when to die. So what are the aspects of light that they're paying attention to? Well, they absorb two different ranges of light. We're talking about red and infrared. Infrared light is absorbed by the battery and it increases the battery charge. They are a battery. They've got a membrane charge across them. And if you expose them to longer wavelength of light, 660 to around 900, the battery on the charge goes up. So in long term, if we actually look at a whole group of mice, extend mice to about 2 years old, 80% of mice will survive to 2 years of age if they're given an environment in which there is infrared light. And you can see this mouse here, quite pucker, quite healthy. If we look at the other end of the spectrum and we look at short wavelength light, particularly 400 to 450, which, we'll go back and we'll emphasize yet again, their wavelengths of light strong in LEDs. You discharge the battery. So you produce less ATP, you've got less energy for moving around, you've got less energy to do almost everything. And if we take the same group of mice and we expose them to those wavelengths and we look at 2 years, only 65% of them survive. So remember, mitochondria control aging and they control the time when you're going to die. Mitochondria are implicated in a vast array of diseases, particularly diseases of aging, diabetes, Parkinson's disease, and dementia, all of which are very significant diseases in Western society with an aging population. So paying attention to mitochondria is super important, and mitochondria have suddenly become incredibly trendy, and that's appropriate. So don't pay too much attention to this, but I just want to tell you that mitochondria do some really odd things. We've got a window on them. We can watch what they're doing because, like us, mitochondria change color when they work. So when you run up the stairs, you get red. Well, mitochondria do something fairly similar. If you're asking your mitochondria to work super hard, You're drawing ATP out of them. They change color. Now, their flexibility, we can watch how flexible they are, because they're nowhere near as flexible in me, well over 50, than they are in young individuals. Now, one thing, a piece of added information to you that may not be obvious. There's more mitochondria in your retina than any other part of your body. Your retina has got the fastest metabolic rate in your body, burns more energy than anything else, and it ages faster. The reason it does that is because you can happily see changes in illumination over 9 log units. Now, to do that, mitochondria have got to work very, very hard. And the worst thing on the planet you can do to a mitochondria is turn the lights out. When you turn the lights out, the mitochondria have got to turn up the amplifier, because they're saying, where's the signal? Where's the signal? Okay, so mitochondria in the retina in the dark are super stressed. When you wake up in the morning and you open your eyes, really the mitochondria breathe a sigh of relief. So let's just watch them breathing. Okay, so if we look at this figure here, here's young people, here's old people. And we're shining a light into the eye, and we're measuring the reflection that comes back. We're not touching the person at all, just asking them to keep their head still. And here we turn the lights out in this period, first 3 minutes. And if you're young, your mitochondria respond very positively. If you're old, your mitochondria just go soggy. So we can watch.
Glenn,
yeah,
we're not quite seeing your mouse movements. Are you able to? So I don't know if you can wiggle your mouse a bit, and maybe we can see your.
Can you see
your
that?
mouse.
See that?
No, so you're off to the side. You're now we can see you. Now we can see you just
Okay.
about. Yeah.
Right. Okay. So this is the old. This is the young. Mitochondria change color depending on their workload. We can watch them. That's really important. So we can watch your mitochondria as you change the lighting in a building, and we can see how they respond. So here's an older person here. Here's an older person, saggy mitochondria. This is an older person we've given a long burst of red light to. Their mitochondria have changed. Right, so mitochondria are watching light. They're responding to light positively and negatively depending on the wavelength. But we live in an environment of solar light where the blue and the red wavelengths have had a stable balance for billions of years. Let's look at that stable balance, and let's look what it does. So this is me lining people up at the back of the Institute of Ophthalmology in sunlight. And this roughly is the spectrum of sunlight that we are measuring in blue. And I'm sticking a radiometer on their backs and a spectrometer on their backs. And I'm measuring the light coming through their body. What light comes through their body? Well, basically 700-800 nanometers, deep red wavelengths, which Bob has talked about. And 99% of those long wavelengths that hit your body are absorbed in deep parts of the body, liver, heart, brain. Bob's pointed out bone is not a barrier. So there is an enormous amount of energy entering the body in daylight. It's being absorbed, but we can measure it just coming out the other side. If you look at this, the axes here are very, very different. So the amount that we're measuring out the back is very, very limited. So again, Bob has told you the bottom two curves represent our sunlight, which we experience outside, and now our light that we're getting from devices like LEDs. There's almost no long wavelength of light. That means that long wavelengths of light are not being absorbed deep in your body in key organs like liver, heart, and brain. The metabolism of your body is changing drastically as a consequence of the fact that those long wavelengths are not hitting deep tissues. Now, I'm going to walk around assuming that all of my colleagues and all of the people I work with are people who are living in LED dominated environments and are not getting really enough infrared. And that is the basis for a series of experiments. And what we're going to do is we're going to stop looking at mitochondria and we're going to say, what are the consequences for the human for having an environment where there isn't much light? I'm going to play to vision. I'm a vision scientist. We've got lots of mitochondria. And the thing about vision is you can measure things. Can you see it? Can you not see it? Is it blue? Is it red? So here is one of our first and rather telling experiments. We take a group of people and we show them a series of letters. And if what we do is we degrade the image so it becomes harder and harder to identify the letter. And we find your threshold. Where's your threshold? Where's the point where you can say, got it, I can get it, but if you make that slightly harder for me, I'm not going to get it. So we take them and the black dot is their threshold when I bring them into the lab and say, can you see these dots? And we've got an array of ages and we've got an array of black dots in these people. Now, we then give them a burst of 3 minutes of long wavelength light. In this case, it's 670. 670 has been popular in experiments for ages. We bring them back and we re-measure their threshold, and that is in black. All the thresholds are reduced. Your ability to see the contrast of colours has been significantly improved. Remember, your retina, more mitochondria than anywhere else, stressed metabolic environment, very, very difficult environment. And we find we can reduce your threshold by 17%. So you've got 17% improvement in the population for detecting colours and their contrasts. Now, interestingly, there's a difference between blue and red. Your blue channel in your visual system is incredibly vulnerable. It's incredibly weak. It goes with aging. It goes with most diseases long before your other colours do. And many people argue you shouldn't see blue anyway. And for that reason, we can improve the blue channel by 17%, but the red channel maybe only by 12%. So this is a clear example of where we use wavelengths of light via an established mechanism, which is the mitochondria, to improve physiological function in us. So we can improve physiological function, and I've been shining lights in the eye. But we've known for some time that mitochondria talk to one another. Your mitochondria in your left toe is rather aware of what's happening to the mitochondria in your right earlobe. They talk to one another. And it was discovered a long time ago that if you disrupt mitochondria in one part of the body, you get a change in another part of the body. The radiologists knew about this long time ago, and it's called the abscopal effect. So if you have a tumour in your right shoulder with secondaries across your body, and that primary tumour is irradiated, your secondary tumours stop for a while. They retract briefly. In some cases, they retract quite a long while. The mechanism is almost certainly something in the blood, and the mitochondria are responding appropriately. So here's a terrible graph, but we're measuring in the blood here something called cytokines. They're inflammatory markers, but they do so much more. And there's a whole range of them. You buy kits to measure them. And we're measuring the cytokines really here in ordinary mice, which is the black histograms, how they change across the body. Then we give the animal a burst of light, and the cytokines, burst of red light, the cytokine distribution changes. So we know that one of the issues that is going on is that there's a messenger system across the body, so that maybe we can shine light at one place and look at an effect at another place. And this has become really quite a big issue. So Parkinson's disease is a mitochondrial disease. Mitochondria in a small nucleus in your brain called the substantia nigra give up and they die. And the consequence is that they are major players in motor control. Consequences, you start to have shaky movements or you finding it very difficult to move at all. So many of the Parkinson's models have been used in primates, and in one of those models, the long wavelength light was shone at the foot, and the motor control was looked at in the hands. And there was a 70% improvement in the hand symptoms as a consequence of the red light being shone on the foot. This started off a whole sort of way of thinking in my lab that I know I bored some of you with before, which is we're saying, right, when you go to get your money, a cash machine, you put your hand as an identifier on a panel, and it identifies in deep red, which you can do, and that delivers a dose of light which will actually improve the physiology of all of your body while you're grabbing your money and you take it out. Now, we treated that as a real joke in the lab, but the issue is it's not a joke anymore. We know that the systemic effects of different wavelengths of light are transmitted through the body. And here's a good example of that, which is an experiment from my lab. And this is very much thanks to Hawley, because I dragged in a number of their people in their lighting team for being subjects in this experiment. So I don't want to fill your world with red light because it's going to disturb you. You don't want to sit at work and have a whole load of red light. I mean, it's going to just disturb your function. So we're going to improve metabolism without seeing the red light. And so in this case, what we're going to do is we're going to shine 850 nanometers at you. Now, your visual system is tuned roughly to 400 to 700 nanometers. I give you 800. If I make it super, super bright, you can just about see it. And in this case, what we did with the lighting people at Hawley and with other people that we brought in is we shone the light on the back with the people facing away. Then I was getting a bit nervous because I realized that the infrared light was bouncing everywhere. So we grabbed a big roll of kitchen film, aluminium foil, and we wrapped people's heads in it. So there was no way light was passing into the eye. So if we take them back and we measure their ability to detect color contrast, we find that if we're looking at the red, remember the red is, we don't pick up red quite so easily, we find an 8% drop in the threshold. If we look at the people with their heads wrapped in aluminium foil and they were still breathing, we get a reduction, but it's not significant. If we now move over to the blue system, testing our blue sensitivity after irradiating our backs with the 850 wall panel, we find a 16% reduction in threshold. That's big. So their ability to detect colors in blue is very significantly improved. And we also find a 7.5% reduction in their thresholds for blue. So we're excluding light coming in through their eyes. At this time, we didn't know how light moved through clothing. So we actually asked them to lift their clothes up and put their backs to the wall panel. But we can, as a consequence, use light to change metabolism for the good in people with light that they cannot see and is not going through their eyes anyway.
Glenn, before we move on to the next slide,
Yeah.
Kathy has asked, is it possible to assess the health of a person's mitochondria?
Indirectly. I mean, yeah, I can with that probe where we shine light and we look at the color of the mitochondria. We can do that way. It's something that industry is taking enormous interest in, and I'm working with biomed engineering at UCL to do that, because I want to be able to walk you into an outpatient clinic, and you could be there for rebuilding your jaw, and you could be there because you've got I don't know, some desperate disease. We want this as a metric, so you walk through the door and whatever is wrong with you, people can say, look, this person's mitochondrial health is A, B or C. I mean, COVID, mitochondria took a real hammering. So yes, it is possible. It's an engineering thing that we're working on. It is conceptually perfectly possible. It just takes a bit of money thrown to it, and I hope we'll be able to do that by the end of next year.
Thank you.
Okay, so mitochondria, let's think about aging diseases. Let's think about diseases that affect all of us. Mitochondria control metabolism, but they need things as well. They produce ATP, but they need their own petrol, and their petrol is sugar and oxygen. So this wasn't my idea. I wish I could claim that it was, but a colleague said to me, well, if we drive mitochondria very hard with red light, they're going to need more sugar. And the only place they can get more sugar is from their serum. I went, yeah, fine. I just didn't get it. But then he said to me, well, do you realize that we could potentially regulate diabetes that way? So this is a very difficult experiment, because my big problem in life is getting subjects. And if you want to get subjects for a really difficult experiment, they don't like it. So if we want to test your blood sugars, we have to get you to drink a vast amount of glucose. We have to prick your finger every few minutes and take a blood sample. And if we're going to do it properly, we're going to have to stick a tube up your nose to see how much CO2 you produce, because if you're burning extra oxygen, you will produce more CO2. But it was in many senses the perfect experiment. So you have a large amount of glucose, and if you look in the green, your blood glucose levels peak at 40 to 60 minutes after you've drunk a vast amount of glucose. That's fairly standard. If you have your Sunday lunch with loads of roast potatoes, your blood glucose levels will peak at around 40 to 60 minutes, and then they go down. If you've had a large burst of red light beforehand, when I say large, it was about 15% of the body surface we irradiated. It was a bright red light. But remember, the message goes round. If we do that, your blood glucose levels do not peak in the same way. Now, the problem with diabetes is not your level of blood glucose. You can have very high levels of blood glucose and you'll survive. What you can't survive is the constant high peaking of blood glucose every time you eat. So we can control your blood glucose with light. And as we controlled your blood glucose, so we increased our carbon dioxide output. So you as an audience, dealing with architecture and dealing with light, actually have the capacity to produce lighting systems that can control metabolism and can control key parts of metabolism, such as blood glucose, which we now know is an absolute killer in an aging population that eats very often crap food, but critically, they're sedentary. You can eat crap food, and as long as you get up and walk around a lot, you can deal with this, but people don't do that. I apologize if you've seen this slide before, but God, I'm really happy with it. If light is controlling metabolism, then can we use it in specific diseases? So, and do we know when we're using light that we're using it where we think we're using it? So there are some very unfortunate kids and they have got mitochondrial disease. Mitochondrial disease, you can't produce enough ATP. And the consequence of that is you lose control of your muscles. So one side of your face droops. You can't control your eyelid muscles called ptosis. So your eyes are half closed. You spend a lot of time in bed and, you know, things are pretty grim. A lot of these kids die, you know, late teens, 20s, they just go into heart failure. So we had a clinical trial, which is still running. It's really difficult to get these kids. There's not a lot of them. So this is Eva, and she had very bad, we were just looking at ptosis at first. She had very bad mitochondrial disease. We started treating her here. So we gave her a torch to shine into her eye that was giving red light because we were interested in our primary metric. Can she open her eyelid? That was the main thing we were going for. Same with Max here. Max had a problem opening his eyelids, although he was a lot more mobile. He wasn't hit as badly by the disease. We got what we wanted. Not only did we get the fact that Eva improved her ptosis to, I mean, she became normal in terms of ptosis. Remember, mitochondria talk to one another across the body. She also gained symmetry of her muscle control across her face. And although this picture is her in bed, still all wrapped up, her mobility has improved enormously. Now, we've not cured the disease, because if we withdraw the red light, we withdraw stimulating the mitochondria, pushing them really hard because they're not good at making the ATP, they will drift back. So, you know, get a bad cold, they drift back. But we can always bring them back. And let's hope that with this case, what we can do is we can get them over the age of 25 and 30, and we can actually maintain a reasonable degree of life expectancy. So, all right, what is our world like in terms of red light? And what can we do about it in a practical sense? We've got a fantastic opportunity at UCL because we've got two really stonking big buildings with no windows. And we can see here an example of it. These buildings, this one is here east. It contains two main things. It contains some heavy engineering, big bits of kit, but it also contains where architecture make all its models. So there's a big area with technicians, and they are sitting in front of their computers, doing things, moving around, but they don't see sunlight. So we've taken a very sensitive infrared camera, and it's brilliantly lit all the time with some really beastly LEDs. And we take a photograph with the infrared camera, and it's black. You cannot see a thing. We measure the color contrast ability of these guys in this environment. It's not great. So then we move in and we take them some 60-watt incandescent light bulbs, and we stick them in around their bench area. They don't have to stay there. We left them there for a couple of weeks. They move around. They do what we want, whatever they want to. And remember, the incandescent light bulb has got oodles of infrared in it. We go back 2 weeks later and we find a 20% improvement in their ability to detect color contrast. Even they were noticing that there was an improvement. We took the lights away, which didn't make us popular, and the effect lasted 2 weeks, and we're still testing it week after week to see how long the positive effects last. So we can go from the lab shining lights in people's eyes. We can go and, you know, into the clinic, and we can shine lights in people's eyes that are pretty sick to stimulate their mitochondria. But this, in many senses, is the bigger issue for public health. We can change the light bulbs, and the consequence of changing the light bulbs is that people's physiology, their visual performance improves, and it's cheap. So
Glenn, sorry, just Dorrit has just posted in the Q&A that she has a friend whose child has mitochondrial disease, and she'd love to get your details. So after the talk, are you happy for me to introduce you to Dorrit?
Not only would I be happy, I'd be over the moon, because we are really finding it difficult to recruit people. So yes, please do, and I would love to have an email from her. All right?
Brilliant. I'll connect you after the talk.
Right. So let me just go. There are some things we can do. We can do these things. We can improve vision. What I really want to do is I want to improve health span. I can't do that in humans because I won't be around to see the end of the project. I can't do it in mice because UCL charged me £42 to buy a mouse, and they charge me 45 pence every day to keep it. And mice in the lab live for 2 years. I mean, I'll be a broken person. But here's an experiment that we did some time ago for slightly different reasons. Bees. Bees are a major element in the agroeconomy. They're very important, and they're suffering because of insecticide that specifically targets mitochondria and gives the bees Parkinson's disease, and they can't move, and they don't eat, and they die. So here we've got a group of bees that have been poisoned with the insecticide, and they're dead at 20 days. If we pair the insecticide with the light, with red light, we find that they perform normally as do the controls. They have a normal lifespan that goes to about 30 days. Okay, so we can give them a Parkinson's disease, we can cure that problem. They're not happy in the first 24 hours, but we can basically cure that problem. But let's have a look at the red line at the top. These are bees that just had red light, nothing else. Look at how many are alive at progressive days. This is the number alive at progressive days. By giving red light, we are increasing average lifespan. They live longer. Remember, there's mitochondria regulate how you age, and it's encapsulated in a mitochondrial theory of aging. And what you find in a bee, you find in a mouse, and you find to a greater or lesser extent in a human. And this has had big economic impact, because in Northern California, they have got rid of nearly all their pollinating insects through very aggressive insecticide use. So they truck up from Texas and from Florida thousands of beehives, and those beehives then pollinate and they come back to Florida and Texas, but 50% of the beehives are dead. Now, this business is run by accountants. It's not by people that love bees, and that's bad news. So now these beehives that are being trucked up and trucked back have got inside them small inserts with long wavelength LEDs, very, very cheap, and it looks as if they're working extremely well. So that's to some extent how we use red light. We use red light to improve things. Can we just go over what happens if we don't do that? If we just say, let's stick with what happens with 420, 450 nanometers, strong wavelengths in LEDs where there is very, very little red. If we look at the graphs over here, this is a lovely experiment everyone seems to have forgotten about. They cannulate people so they can monitor their physiology, and then they turn on, for this period, a blue light at 420, 450. And this is your blood pressure. The blood pressure instantly drops when the light goes on. And this is not a bright light. This is a light at what we would consider acceptable level. And as long as the light is on, the blood pressure drops. If you look at the heart rate shoots up, which you might expect that with blood pressure drop, but the heart rate shoots up. So walking into a room can have a profound effect on your physiology, on your cardiovascular system, just by the 420, 450 nanometer light. And as Bob has pointed out, the astronauts on the International Space Station, which are under this light for a really long period of time, we think their mitochondria is slowing down. NASA called it a mitochondrial problem. They were certainly right. So these astronauts are becoming pre-diabetic and they're aging faster. That's a real, I don't know if anyone remembers the end of 2001 where the astronaut ends up somewhere where he starts getting younger and younger. Well, they probably got that the wrong way around, because in actual fact, you're getting older really fast. These experiments meant that I stopped working on blue light and humans, but other people have done some fantastic things very recently. If I slow your mitochondria down, they're not taking out as much glucose from your blood. Where does the glucose go if it's not being taken out? Well, these are mice that are either under 420 or 450 nanometer light. They're young mice. This is their weight. What happens? They get fat. They get very fat very quickly, and they lose control of their glucose metabolism. So these lights, we're not talking ultraviolet, we're talking lights in LEDs.
There's people, oh yeah, no, the slide hadn't changed for me, that's what I'm saying.
Okay, so, but it does more than that. Let's go to behavior, because behavior is the real big one. Very often people kind of want to know, how do mice feel about their environment? So they drop them in a box and they film them from above. They film what they're doing. And if we do that to a mouse, what you find is you get an anarchistic pattern. Mice are wandering all over the place and looking around. And this is 420 and this is 450. So 420, oh, it's both 420, sorry. They wander around, they explore their environment, they're perfectly okay about it. If those animals have been exposed to short wavelength light for a period of time, they become incredibly cautious. A nervous mouse will not walk to the center of arena. It only hugs the walls. If you've got mice in your house, in your kitchen, you'll know about it because mouse poo right up against the base of your units. It's a new environment, they don't like it. So these mice behave in a very cautious manner, and that cautious manner is consistent with low level infection. When you've got low level infection, you don't go around exploring things. You actually take a very, very cautious stance. And remember, longer wavelengths reduce inflammation because mitochondria control that, and shorter wavelengths that undermine mitochondria push inflammation up. So I'm going to come to the same conclusion as Bob. Bob came to it more from a theoretical point of view. I'm coming to it much more from, I suppose, a pragmatic point of view as an experimentalist. We're not going to argue about the fact that daylight drives healthy physiology. We all know that. We're not going to argue about the fact that LEDs cut most of the visual solar spectrum out. That's very clear. If we sit down and look, we're also not going to argue about the fact that a large number of epidemiological studies show repeatedly that mortality rates for a lot of diseases are much lower in those that experience significant sunlight. And mortality rates are higher than those that do not. And in that category, I will put people who spend a lot of their time in the built environment. So one of the issues that we have to face is changing lighting, and that issue is a matter of public health. So when I go down the pub now and, you know, someone says, well, what do you do? Well, I don't say I'm a visual scientist anymore. I kind of tend to describe myself as a public health light person. And when we consider it, there are so many different angles. We've got sunlight, we've got our LEDs. LED lighting is horrendous in motorways. It's horrendous in floodlights. The energy that's being kicked out at 420, 450. I've used the example many times. The worst source I've found is the BMW laser light on a car. You can see for miles, but it's an absolute killer. We still very happily put our babies under quite intense blue light if their liver isn't functioning perfectly well when they're born and they're producing too much bilirubin. It works for the bilirubin, but why we're doing that without, and the baby here has got its eyes closed. No one's told the physicians that mitochondria respond to light across the body. And the one thing I've not talked about, because it's a little bit tangential, is the distribution of light in the built environment with the loss of red is now recognized as a driving factor in myopia. In myopia, which is it's not a ticking time bomb in the Far East now, it is a bomb. In myopia, the eye grows longer. As it grows longer, the retina tears. As the retina tears, the center of the retina degenerates. I've not talked about myopia because even though the evidence is absolutely clear, I don't quite understand the mechanism. But there are multiple reasons here why changing the light bulbs, as we did for those guys here east, can have a profound effect on physiology and take away some of the problems that happen in health span, particularly as we age. Thank you.
Thank you very much, Glenn, and also thank you very much, Bob. I don't know if anybody's got some questions following both of those quite fascinating talks. We've got somebody who's just done some clapping hands. Not sure who that was, but thank you very much. But yeah, if anybody's got some last-minute questions that they want to throw out there, then please just drop them into the Q&A. Here we go. We've got one just come through. This one's from Charlotte. Outside of changing bulbs, are the red light products sold on the market for health and well-being generally effective at the moment?
I'll be clear about that one. Most of them are crap. My outer office has got loads of them. People send them to me to test. Most of them are the wrong wavelength. They're the wrong energy. A lot of the ones that come from China are terribly unreliable, and there's a product recommending for your eyes. When you turn it on, we found in about 2 or 3% of them, they just get very hot and they go pop. You have to be so careful. There are, I think for eyes, I'm going to say just be super careful. There's one company with who I have no commercial interest, who produce products that we use, and I think they trade under the name now of Light Power Health. We've used their goggles in clinical trials. We've used some of their lamps, but most of what you see, even if it's good on the day that you buy it, they use terrible LEDs and 3 months later, they're not doing what they should be doing. So I can only say, be very cautious.
And that's, I'm assuming you're referring to sort of the terminology that I know you don't like, which is photobiomodulation.
That's right, because there's too many syllables in it. So Bob came up with this new one, photometabolism. I can't say I'm a great fan of it, but that's a hell of a lot better.
Bob, you had your hand up.
Can I comment on that question as well?
Yes, for sure.
I do very practical things in my house. I have done for a long time now. I mean, I do have red LEDs, which we switch on every morning, 660 and 850 nanometers. They seem to be reliable. But what I do in the kitchen and also in other rooms, our kitchen has white LED downlighters. They're 3000 Kelvin downlighters, and they're pretty horrible to live in, but you know, they're there and they help you read. But over the kitchen table, we have a set of 3 nice sort of saucer-shaped bulbs with covers with quartz halogen bulbs, which we bought in Germany about 20 years ago. And we installed them back here. And I run those quartz halogens at one stage below the maximum brightness. There's a sort of digital dimming process. So they run at a lower color temperature. The bulbs have been in there for nearly 20 years, never been changed. When I measure the spectrum of the light hitting the dining table, the combination of the white LEDs and the quartz halogen bulbs matches the sun, as well as that example I showed you on one of my later plots. And it actually cost me nothing. So the combination of lower voltage, ordinary tungsten filament lamps or quartz halogen lamps, doesn't matter, run them at lower voltage with a simple dimmer, and white LEDs, pretty good.
And yeah, maybe the concept of using a filament as a heat source rather than a light source could be valid as well, right?
Well, I mean, I cut myself off when I was going to talk about that a bit, but I suggested it to the local Eon heating engineers last night, actually. If you're going to heat a building, why do you heat a building? You don't heat a building for the building's benefit necessarily, although you might want to stop the water freezing. You heat a building to make the occupants healthy and comfortable. Now, if you use electricity to heat radiators, water radiators, say to 50 degrees or something, 60 degrees, that's an incredible waste of a high quality energy source. Why don't you divert some of that to making infrared light at which is effectively just over 2000 degrees, and shine that infrared light on the occupants. It'll go through the occupants, it'll make them healthy, and it'll be excreted as heat. And you won't have lost any energy at all. You won't have had any extra energy in your building budget at all. But you will have used some of that light intelligently to keep your occupants healthy. You know, to me, it seems a no-brainer. You'd never want to use a high-quality source of energy like electricity to produce low-temperature heat. If you can produce high-temperature heat and use it for biological health.
Martin Morreed has just posted a question. What types of heaters emit 850 nanometers infrared? Does my oil-filled electric heater emit this IR?
I have no idea what the answer to that is.
Let me answer that. A flame glowing yellow, like a candle flame, or some kinds of, I guess, oil lamps will, as long as the temperature is, say, between 2000, the color temperature is between 2000 and 2500 kelvins, then you're producing near infrared. If it's cooler, you're producing further infrared light, but not very much near infrared. So the glowing embers of a coal fire won't produce very much near infrared. But when the wood burning stove is producing yellow flames, then you've got quite a good flux of near infrared radiation.
So Brian H just messaged saying, truly fascinating. I learn more every single day. Thank you. And then, our friend Scott in New Jersey has just mentioned that optically children are more impacted than adults.
Definitely.
More of a statement, not so much of a question.
Okay. Well, just think about it this way. Think of the thickness of the tissue. Tissue density in a child is far less than tissue density in a human. In an adult. So I would say children are much more affected, and also they're growing. God knows what you're doing by blasting them with loads of blue light that close down their mitochondria. Definitely I would be extremely concerned about exposures of short wavelength light to kids.
And then we got an anonymous question. Does sun cream negate the benefits of spending time in daylight / sunlight? So I'd sort of probably suggest that if we're talking about ultraviolet and the production of vitamin D, then maybe yes. But what about infrared? I'm assuming that infrared will just still go through.
Exactly. Sunscreen will have very limited effect, if anything, on longer wavelengths.
So, in your next talk, Ulis, Scott will tell you about sunblocker.
I'm sure he will. I'm sure he will.
Yes.
And then when we get the chance to talk with Richard Weller, then I think we'll go into even greater depth on that front, on a dermatological perspective, which I think is going to be fascinating. So very much looking forward to those. It's
a complex story, and I think they're changing sunblockers now to include blue as well as UV, but it's not a nice story at all.
No. So we have no more questions coming up at the moment. So Glenn and Bob, I'm going to thank you both very much for your time. It's been absolutely fascinating. I'm sure those who are still online will agree. And as we just mentioned, that we've got Scott Zimmerman joining us in December, where we'll be talking about melatonin, light, and the optics of the human body, which is going to be, again, absolutely fascinating. And then we'll get ourselves ready for the future talks in the new year. So, thank you very much, both. And thank you everyone who joined. Have a wonderful evening. Oh, hang on a second. We've got. No, okay. Andy Ball saying fantastic. Thank you. So thank you very much, Andy, for the comment. And Rob Mayer, both talks so fascinating. Many thanks for the unique opportunity. So thank you very much, both. Thank you for your time, and I look forward to catching up with you in the not too distant future. And the opportunity to learn from you is always just amazing. So thank you.
Cheerio.
Thanks a lot.
Thanks a lot.
Okay.
Thank you.
All the best. Cheers.
Take care. Bye.